now we're very lucky. We have many companies who are working on Pompe disease. And I decided that if I stop talking right now, which I will, I can save some time for people who are presenting, because the timing was limited to 10 minutes. So if I'm not going to talk at all, then I can allow them to use extra 117 seconds, please.
Wow, thank you. So thanks, Nina. It's really an honor to be introduced by you, for sure. Thanks to the organizers, to Tiffany, to all of you. I've presented in front of some of our clinical experts, but I haven't presented in front of our patient experts. So it's a little nerve-wracking, but also a little exciting. So thanks so much for coming. So today I'd like to introduce you to Amicus and give you some updates on a treatment option for some patients with LOPD. I have 10 minutes, 5 objectives. 1, introduce you to Amicus. 2, to try to explain a little bit about why companies like Amicus and our friends and colleagues here thought that even though there was an existing treatment available, it was still an opportunity to develop some other options. 3 and 4 kind of combined the approach that we took to try to solve some of these challenges or maximize some of these opportunities, and tell you how we did by reviewing some of the study results. And finally, a look at the future, what we have going on at Amicus. So on the next slide, I just want to introduce you. Oh, sorry, I guess I have this here. Thanks. Okay, so Amicus is a company, so we're focused in rare diseases and aim to provide support for communities living with rare diseases. It's a global company committed to developing and advancing treatments for rare diseases. It was founded in 2002. And in addition to the Pompe therapy, which I'll talk a little bit about in a few minutes, we have another approved treatment since 2018 in another lysosomal disease called Fabry disease. So a little bit about Amicus. Our inspiration, again, is attending events like this really reminds me of what we do and why we do it, just providing support for the rare disease community. This foundational belief, our zeitgeist, if you will, comes from the family that you see on the right there. That's the founder and former CEO, John Crowley, his wife Eileen, and his 3 children, 2 of which live with Pompe disease. And I think if you come to Amicus, I hope that you'll see that there really is a genuine belief. I think John's vision has remained true. I think I have colleagues here and back at the office who I think really do live this every day, and I think it's something that we really feel is important, and it's really a great place to come to work. Our current CEO, Bradley Campbell, President and CEO, I think outlines our vision for the future as a company. Again, we'll always remain in rare diseases and in building in the patient perspective into everything we do. And I think our patient advocacy team, patient and professional advocacy team, do a remarkable job of working with the community, the patients, the caregivers, the patient advocacy organizations to bring their needs, their insights into the development and the development process. We'll continue to grow ideally, and I think everything that we would do, if we invest in a new disease or a new treatment, a new disease state, it will always be a rare disease, a devastating disease, one that we feel provides an opportunity to create a first-in-class medicine, and one that the clinical data actually can demonstrate the benefits to those patients. Okay, so that's introduction. That's objective number 1. That's Amicus. That's me. But a little bit about late-onset Pompe disease. I don't think we need to spend a lot of time here. Many others have gone through this. But if we look at a hypothetical cell, a cartoon of a hypothetical cell on the left, this is what good looks like. There's lysosomes that contain glycogen within the muscle cells. There's an enzyme in there, ideally, that can break down this glycogen. But in an aberrant state or in a disease-causing state, we see what not so good looks like, and that's what's shown on the right. So we have an increase in glycogen content. These lysosomes no longer look very healthy and nice, tight circles, but have sort of become disrupted. Some of that glycogen is leaking out into the cell itself, And this obviously ultimately causes muscle damage. So thankfully, LOPD, there are treatments available so far today. All are based on at least have a component of ERT, so that's the enzyme replacement therapy. So the cartoon on the right, you see the muscle fiber. You see that receptor, which is really, we're very fortunate that we have evolved to have these receptors on the outside of cells to allow for enzyme which is administered into the blood. The little that makes it to the muscle interface has the opportunity to bind and get into the cell where it can hopefully degrade that accumulated substrate glycogen. So while there is a lot of little circles in the blood, that ERT, you can see that only very little makes it to the muscle interface. So we heard a little bit about L-glucosidase alpha, a life-saving, a life-altering treatment for so many patients. But we also saw that there's some waning efficacy over time in some patients. And some of the reasons for that come down to this process, this long journey from the blood to the muscle to the lysosome where it can actually function. And that's sort of the foundation for Amicus's approach, which I'll hopefully describe in a few minutes. So since very little makes it to the muscle interface, we're going to talk about things like adornment or pieces of the enzyme that help bind to the receptor. We're going to talk about processing as the enzyme makes it into the lysosome where it can be most active. And we're going to talk a little bit about miglustat, which is a small molecule that can bind to and stabilize those blue circles that you see in the blood to hopefully drive a little bit more enzyme to the muscle cell. So I'm going to introduce you to Pombiliti Opfolda. So Pombiliti is the recombinant enzyme. Opfolda is a small molecule stabilizer. It's a little tricky here. We're an international conference, but we're focused in the US. So what I'm going to talk to you today is about, based on what the FDA tells us, we're required to say what we want to say because we're in the US. So the indication in the United States, as others have mentioned, is for patients who are on ERT and not improving. They must be adults. They must be at least 88 pounds. So like other ERTs, there are some important safety considerations. There's a risk of hypersensitivity reactions. There's a risk of infusion-associated reactions. And there's a risk of acute cardiorespiratory failure in some susceptible patients. It's really important that we mention the indication, but also the contraindication. So pregnant females should not be taking Pombiliti and Opfolda. You're going to talk to your healthcare provider about any medical conditions, particularly those females who are looking or may want to become pregnant. These two medicines have only been studied together. They must be taken together. Pombiliti and Opfolda must be taken together. There are some common side effects. We actually have a slide toward the end where we'll have a look at those, but look somewhat similar to those that you've seen for other approved medications. Okay, so our approach, number 3 and 4, how did we do? So Pombiliti, as I mentioned, is an enzyme replacement therapy. We talked about binding to the receptor. L-glucosidase alpha has a proportion of those enzymes have the right structure on the outside of adorned on the enzyme to allow for uptake into the cell. If we could find an enzyme that had more of this ligand for the receptor, then we might be onto something. We might be able to drive uptake into the cell. And so Russell, who's going to talk to you about something else, and Hung, our former chief scientific officer, really did a remarkable job of looking for an enzyme, a recombinant enzyme with certain characteristics to help drive that uptake into the muscle without the need for any kind of changes to the enzyme after it's manufactured in the Chinese hamster ovary cells so that it can then be processed into the most mature, most active form of the enzyme in the lysosome. Because at the end of the day, what do we want to do? We want to get as much enzyme into the muscle, into the lysosome, make sure that it's as active as it can possibly be so that it can degrade glycogen. And that's really the foundation for this 2-component therapy. So at the end of the day, it's the enzyme that's doing the work. The enzyme is degrading the glycogen, but it's also administered with this small molecule. And the rationale for that small molecule comes down to what somebody had mentioned yesterday, I think it was Professor Roberts, that these are lysosomal enzymes. These are enzymes that function best in the acidic pH of the lysosome. They don't like, or they're not as happy in the blood. They can be degraded in the blood. They can be denatured in the blood and inactivated. So if during that brief period of time where the enzyme is in the blood, we can bind it and stabilize it, then we can drive what's available for delivery to the muscle cells. So that's what we've done by using Opfolda. Again, they must be taken together because they've been studied together, because they're designed to work together, and we've looked at the indication already. Dosing, like other enzyme replacement therapies, is weight-based, so you'll work with a clinician if you decide to use Pombiliti and Opfolda. So here's an overview of how it works. I mentioned this journey, so it's fantastic that we can have the technology available to generate recombinant enzymes. It's administered through the blood on the left-hand side, but at the end of the day, it has to get all the way to the lysosome, which is on the right. And that journey is not as easy as it sounds, right? We know which enzyme is not working well. It sounds so easy. We could just replace that enzyme and we can make patients better, but it's a little more complicated than that. So what you see here is a schematic of these two components. The pink structure is Pombiliti. That blue octagon is the Opfolda. They bind together. They travel to the lysosome where some additional things happen, and we'll go through each of those steps now on the next two slides. So in the blood, Pombiliti is bound by Opfolda. It's protected, it's stabilized during the time that it's in the blood. It now can enter into the lysosome. In the lysosome, again, in individuals living with late-onset Pompe disease, there's an accumulated glycogen. So this entire box here that you see is a hypothetical lysosome with glycogen accumulated inside. Pombiliti and Opfolda can enter the muscle cell. Opfolda is removed from Pombiliti, so Pombiliti can actually do its job, which is to degrade the glycogen. There's one more step, though. You notice that the size of the Pombiliti on the left is larger than what you see in the middle. That's because there are some scissors or some apparatus that help trim that precursor protein, that precursor enzyme, into the most active and mature form in the lysosome. So now the binding site is available for binding to glycogen. That glycogen is degraded into small glucose molecules, and that's, again, the idea behind any enzyme replacement therapy, including Pombiliti. Okay, so now how did we do? How did we do? So we had this innovative design for a treatment, but at the end of the day, the gold standard is a phase 3 clinical trial, so we have to see how did our thinking, how did our rationale lead? What were the results? So we ran a phase 3 clinical study. It had 123 individuals who participated. Am I out of time already? I have 2 slides. Okay. Okay, you'll tell me when. So the interesting part of the study and the important part of the study, as was mentioned, is that it didn't include only naive patients. In fact, only a very small percentage of the subjects were naive coming into the study. It actually was majority, almost 80% patients or subjects who had participated were on ERT for at least 2 years, and actually many, many more years, as we'll see in a second. So 85 of those individuals received Pombiliti and Opfolda. It was a 2-to-1 randomized study. 38 received the comparator, which was L-glucosidase alpha plus placebo. 6-minute walk distance was the primary endpoint. FVC was the secondary endpoint. And as, again, that ERT experienced population, actually patients were on ERT, individuals were on ERT for 7 and almost 7.5 years coming into the study. So let's take a look at the primary endpoint, 6-minute walk distance. And in a way, at baseline, if you think about this, you have these are only subjects who had been previously treated. That's our indication that those are the data that we're presenting here today. Our whole study, that 123 subjects, 6-minute walk distance didn't meet its primary endpoint for statistical superiority. The FDA evaluated that and said, okay, but let's have a look at the ERT experienced population, and again, have evaluated that and approved the medication for use in these individuals. So at baseline, these patients who were on treatment for 7.5 years before coming into the study, either they were randomized to Pombiliti and Opfolda, that's the dark blue line, and after 52 weeks, they improved by 53 feet. So 53 feet is the length of a tractor trailer, an 18-wheeler. If you've seen or driven past one, that's what 53 feet is. Or after 7.4 years on L-glucosidase alpha, you stayed on L-glucosidase alpha. So that group is on the bottom. After 52 weeks, an increase in 2.3 feet from baseline. FVC, our secondary endpoint, is represented here. So again, the top is Pombiliti and Opfolda, those patients coming in for 7.4 years. After 1 year, no change in FVC. If they stayed on L-glucosidase alpha, a decline of 3.5%. Safety reported here. Most common side effects, additional side effects on the right-hand side. Dosing and administration, so we've added on this oral component, this miglustat, Opfolda, which requires a unique dosing paradigm. So the infusion is still 4 hours, but there's a 4-hour fast. You fast, take Opfolda 2 hours into the fast, the fast for 2 more hours. The Pombiliti is started 1 hour after taking the Opfolda. And last objective, tell you a little bit more about what's going on. So we have some clinical trials ongoing. Most importantly, our pediatric studies. One pediatric study called the ZIP study in pediatrics age 0 to 18, and our Rosella study, a pediatric study in IOPD. If you're looking for more information, please reach out to us, the email on the screen, clinicaltrials.gov, our patient and professional advocacy team, sorry, our booth, our table in the other room, we can give you some more information. And last but not least, we're going to have some very challenging conversations with the experts in this room, the advocacy organizations, in terms of creating that real-world data story, a registry. So we're excited for partnering with you all on that. So thank you very much.